Hello, listeners. Yamina here. I'm very excited to have you join us for another Dr. GPCR podcast episode. If you haven't heard, we are officially a nonprofit 501c3 organization. Becoming a nonprofit only solidifies our mission to serve the GPCR community. Therefore, we're taking a short podcast break starting in July to come back with new episodes at the end of August, as well as an amazing set of events in the fall. If you are an ecosystem member, by now you know that every Thursday you get your weekly news or weekly GPCR news delivered directly in your inbox. If you're not an ecosystem member yet, join us today. Thanks to our fantastic team of volunteers of GPCR scientists, a GPCR article will also hit your inbox weekly on Tuesdays. Also, mark your calendars for not one, but two Dr. GPCR University classes with Dr. Terry Kanakin. The first one is titled Techniques for Effective Lead Optimization of Candidate Molecules, Principles of Pharmacology for Drug Discovery, which will start on Thursdays, September 5th at 10 a.m. EST and run for 4 consecutive weeks for 90 minutes. Then, on October 3rd, an advanced class will start about therapeutic translation of GPCR activity, advanced method for the optimization of candidate selection. This class will run for 5 consecutive weeks, also from 10 a.m. Eastern Time on Thursdays. For both courses, we will be offering 5 scholarships per course to GPCR scientists who live and work in developing countries. So stay tuned. Just a short week after the beginning of our second Dr. GPCR University class in the fall with Dr. Kanakin, on October 11th, we will be hosting our Dr. GPCR Symposium on GPCRs as therapeutic targets. During that day, we'll have talks from speakers of all levels, both from industry and academia, a poster session, as well as a panel discussion. We're also excited to be co-organizing our very first in-person Adhesion GPCR workshop in collaboration with Dr. Anthony Boucard from the Adhesion GPCR Consortium. Mark your calendars for October 23 to 25 and join us in Mexico City for a fantastic meeting on all fascinating aspects of Adhesion GPCRs and more. If you'd like to sponsor the Adhesion GPCR workshop, please email us at hello@drgpcr.com. Both emails, actually, we have now 2 emails, hello@drgpcr.com and hello@drgpcr.org. Both of these emails will be active for a foreseeable future to make sure that we don't miss your emails. And now, let's dive into this episode. Hello, everyone. This is Yamina from the Dr. GPCR podcast. And today we have with us Dr. Benjamin Myers. Hi, Ben. How are you?
Good. How are you?
I'm great. So glad that we were able to make it on the early Saturday morning. So tell us a little bit about your current role and about your story as a scientist.
Sure. So I'm currently an assistant professor at the University of Utah School of Medicine in the Department of Oncological Sciences, also affiliated with the biochemistry department there. And that's a role that I've been in since, I guess, early 2018, so a little bit more than 3 years now. And my lab is really focused on trying to understand signaling at the membrane, and particularly how it influences developmental biology and cancer biology. And so we're really focusing a lot on class F GPCRs in particular, in sort of the frizzled and smoothened subfamily, so sort of the odd members of the GPCR superfamily. And I'll say most of our work on this point has really been on smoothened, which I guess if the frizzled, or if class F is an outgroup, I guess smoothened is sort of the outgroup from the outgroup. But a really critical, really fascinating protein, and really trying to understand some of the key roles it plays in development and cancer, but focusing on how it's activated and how it signals downstream to mediate changes in gene expression, and they go on to dictate cell fate. And so we're thinking a lot about these questions about how smoothened is regulated, and also how we can target smoothened in a better way to help in disease. And then another area that we're interested in from our work on smoothened is thinking about GPCRs in the primary cilium, which is this tiny little antenna-shaped structure that a lot of people don't know about, but it's present on all the cells in your body. It's this little cell surface organelle. And it ends up being really critical for a lot of aspects of human health, and also sort of a hotspot for GPCR signaling in the cell. There's a lot of mystery surrounding kind of how signaling takes place there, and that's another topic that we're working on.
That's fascinating, developmental biology. I remember taking a developmental biology course. I think it was during my master's, and I just hated it, to be honest, but I'm glad to be talking to you. Hopefully you'll be able to change my my not so good experience during that course, and especially that it wasn't about GPCR. So I'm glad that we're talking about GPCRs today. Before you got to the University of Utah, where were you before? What led you to where you are today?
Yeah, okay. So maybe I can start. I guess I can start pretty early. So even I guess growing up as a kid, I was always pretty interested in science. I was really curious about sort of the natural world, and especially about, I guess, the human body, and about health. I had some scientists in my family. So my dad is a patent attorney, and he worked a lot on biotechnology cases, and so he would always be talking about science and about sort of things like that at home. And so I kind of heard a lot about that from him. And my grandfather was a chemical engineer, and so he was also very much a scientist. So neither of them were sort of academic researchers, but I think science was sort of something that was always around. And I kind of gravitated, I guess, more and more towards sort of biology and human health growing up. And I guess I always thought that was just a really interesting and a really important topic. And so in college, I kind of had this idea that I was going to become a doctor and go to medical school. But I started working in a lab, and I was actually working with Stuart Schreiber, who's sort of a chemical biology person, doing a lot of different types of science, kind of at the interface of chemistry and biology. And I think the more time that I spent working in his lab, the more I really started to feel like patient care was probably not my calling, but the research and sort of working in science, sort of the intellectual challenge of it, and just sort of the excitement of making new discoveries, that really was more my cup of tea. And so after that, I made up my mind to kind of shelve the medical school plans, and then I applied to grad school instead. And so in grad school, I was at UCSF, and I was working with David Julius, who some of your listeners may know him. He's someone who works a lot on understanding how we sense pain and how we sense temperature, but very much at a molecular level. And so his lab focuses a lot on ion channels that are expressed in our sensory neurons that mediate these sensations. So I guess, for example, if you've ever eaten some really spicy food and you've found that you have this burning sensation on your tongue because you overdid it, it's because there's a chemical compound in chili peppers called capsaicin, which activates an ion channel that's expressed in your sensory neurons that basically opens and depolarizes the neuron. And that's the same ion channel actually that also responds to hot temperatures. So the reason that capsaicin feels hot is because it's activating the same molecule that heat activates. And I just thought that was so fascinating. And I mean, not just the idea that there's this sort of immediate connection between the natural world and the environment and what he was studying, but just the fact that it kind of came down to this sort of really elegant molecular, I guess, machine that was mediating this. And I think it was really in David's lab that I really fell in love with, I guess, membrane proteins, just as these incredibly kind of dynamic and just, critical proteins that in a lot of ways, I guess, mediate they're really what help the cell interact with its outside world, right? I mean, they're the things that help cells sense danger and to sense, environmental cues, and even
Light,
to sense,
yeah.
Light, absolutely. So I really fell in love with membrane proteins there. And I think the other thing that I loved about being in David's lab was that I really learned from him, from the people in his group, a lot about, the value of really getting down to sort of a molecular explanation for things. And that, it's not really enough to just understand what the players in this sort of pain pathway are, but we have to really understand how do they work at a kind of a deep, biochemical and molecular level to really, fully understand how the system works. So that was something that I think, and so in his lab, I was trying to understand basically how these ion channels that sense hot temperatures, and there's another one that senses cold temperatures that responds to menthol, which is the sort of the soothing ingredient in mint. So I was just trying to understand how they work. And so I was doing a lot of structure function studies, a lot of mutagenesis, and a lot of functional assays, mainly patch clamping and electrophysiology. And yeah, I just really loved it. And then I guess the other thing about his lab was just he's a really bold scientist, and I think he was someone that really encouraged the people in his lab to be bold and also to take risks. And it was just a really fun like lab environment to be in. It attracted some really big personalities and people that are kind of work hard, play hard type. And so it was always just fun to come into lab because you never really knew what you were going to find. There was a lot of people in the lab that were working on sensory biology, but in other, non-mammalian organisms. And so, as a way to understand more about how they, how these systems evolved and how they're tuned in different species. And so, you could come into the lab and there might be like a baby alligator in a cage that somebody was working on, or like a rattlesnake or a star-nosed mole, which is this sort of bizarre creature that lives underground that has no ability to see really, but basically it's evolved this very hyper sort of aware sense of touch. So yeah, there's all these sort of these crazy animals, and people were just having a really good time. Really focusing intensely on the science, but also just taking being really really bold and really creative. So that was a really good experience. And then kind of when I got to the end of my PhD, I kind of had a little bit of a crisis because I didn't know what I wanted to do next. And I still really liked what I was doing, and I liked ion channels. And I thought a little bit about, maybe continuing with that in another lab. And, David really encouraged me to kind of branch out and to try to do something a little bit different. I mean, that was something that he had actually done in his career. And it was something that he encouraged a lot of his trainees to do, to sort of switch fields, because, you really get a different perspective coming into a new field from a different field than if you just sort of stay in the same field. And, I took his advice to heart. I didn't really know what I wanted to study. And then I remember this was at UCSF. They had one of these seminar series where they would have, really great scientists come in and give talks, but also talk to graduate students about, how to be a scientist and things like that. And so Christiane Nusslein-Volhard was one of the speakers, and she is a really famous developmental biologist who made some really seminal contributions to how organisms develop with these amazing screens in fruit flies and in zebrafish. And so I remember she gave this talk to the students that I was at. And one of the things that she said right away was, if you're looking to do a postdoc, I would advise you to just completely change your field, because I did that. And it turned out she had actually studied like the biochemistry of DNA replication as a graduate student. And then she just kind of got bored with that and just completely went on to something else. And she said, you guys should all do that too, because it's really invigorating, and you really, really kind of, you kind of get to press the reset button.
Yeah.
And so I was kind of naive, and I was like, oh, that worked for her. So, maybe that'll work for me too. And I started to get really interested in development. I started to read about development, and I just felt like, first of all, this is such an interesting question. How does an organism develop? I mean, what it really comes down to is, we have to basically build ourselves from nothing, from a single cell into an organ, into a human being with all these different types of tissues and organs, and they all have to adopt the right fate, and they all have to become the right thing at the right time. Like you have to, and if that doesn't work, it's a disaster, right? And there's so many things that have to happen. And I just thought, my God, this is such an interesting question. And the more that I started to read about it, I just realized that we actually don't really have any idea about the signaling that allows this to happen. I mean, we know that there's a couple of these really key signaling pathways, but in terms of how they work, it's really, a lot of cases, it's really wide open. And I think this is something that really grabbed me, because I knew from, the work I had done as a graduate student, like how important it was to really get down to sort of the biochemistry and the molecular mechanism, and how that really informed the way that we think about that area of biology. And I saw that there was just a lot of potential to do that in development, in developmental biology. So I started to read more about that, and then I pretty quickly triangulated on one specific developmental pathway called the hedgehog pathway. And so it has this this funny name because it was discovered using fruit fly genetics mainly. But it's this really critical signaling pathway that's basically one of the handful of pathways that is responsible for the development of pretty much every single organ in your body. So in almost every case, you need hedgehog pathway activity in order to pattern those.
That I remember from my developmental biology class.
Yeah. There's a couple of these, hedgehog and Wnt and FGF and Notch, and it's really kind of amazing that there's not so many of them, right? These pathways, and they just get used over and over again in these creative ways, to pattern all of these structures in your body. And so in looking at hedgehog, what I realized was that this is a super critical pathway. I mean, I had also heard of it, even though I didn't know much about development at that point. I knew it was really important in embryogenesis. I knew it was really important in stem cell biology and cancer. But, the more that I started to read about it and learn about it, it just really shocked me that some of the most basic aspects of how this pathway works, people have no idea, or had no idea at the time. And it wasn't like these were questions that were down in the weeds either. It was really like, what turns the pathway on? Like, what transmits the signal, in a cell that where it gets turned on? And I just thought, these kinds of wide open questions are a really great I guess a really great ground for doing a postdoc, because there's lots of stuff to still figure out. And so I did my postdoc with Phil Beachy at Stanford, who's sort of one of the experts in the field in hedgehog signaling, and who had made some really nice contributions in that area. And I started to work on some of these questions, and pretty quickly my interest triangulated on Smoothened, which again is this class F GPCR. And it really is kind of at the heart. It's really at the heart of the pathway. I mean, it's a protein at the membrane that has to be turned on in order to turn hedgehog signaling on, and it has to signal, from the cell surface, downstream into the nucleus. And nobody really knew how any of that stuff worked. And I think the other thing that sort of drew me to that in particular was that it just seemed like the little that we knew about how, I guess, hedgehog signaling generally and Smoothened in particular worked. It just seemed like it was so weird. It didn't follow any of the rules that I had learned about for other signaling pathways. It seemed to just break all the rules. And so I realized that it's a really important thing to work on, but also that it's going to definitely need some time to really crack. So this was something that I started during my postdoc, understanding how Smoothened is activated in cells. Yeah. And that's something that I continued in my independent lab, and also have started to work on some other aspects of Smoothened biology. And then that's also kind of interestingly brought us full circle, and now taking us to back to some more general themes in GPCR biology as well. So yeah.
Wow, I love that you went back to being a teenager and being interested in science. Let's take a step back, because I have a couple of questions for every stage of your career.
Sure.
You mentioned that you were always interested in science, and then you had your father and your grandfather having that scientific mind. Was there a particular subject that you would have preferred? I mean, I think you mentioned biology, but some of my previous guests said astrophysics or something like that. But was it biology for you?
Actually, so my dad and my grandpa, I guess they were really more into the chemistry than they were into the biology. I think a lot of what my dad ended up doing was sort of pharmaceuticals, and so that definitely touched on the biology. And I think for me, I guess the more that I started to learn about the biology, the more I just felt that was really the thing that really piqued my curiosity. Mm-hmm. So I guess that was kind of how I guess it was sort of coming in from maybe the chemistry angle, but definitely like gravitating more towards biology, just because I felt like the questions were things that kind of interested me more.
Yeah, I think chemistry is fantastic, and as in the series Breaking Bad, the chemistry must be respected. But if there's no biology associated to it, then it becomes this abstract topic.
In some ways, yeah, definitely.
It was so fascinating to hear about the capsaicin response, or example that you had, and it actually reminded me of a documentary I've seen. It was about food and food things like that, but basically chickens apparently can eat the red peppers.
Yep.
and people give it to chickens so that their eggs, the egg yolk becomes even more pronounced red.
That's right, and actually there's a reason. So there's a reason for that, which is that basically birds are really good dispersal vehicles for the capsaicin for the chili pepper seed, right? And so the plant has actually evolved capsaicin in a way to ward off rodents like rats and squirrels and I guess us, I mean, human beings too. But not birds, because birds will help to disperse the seeds. And so actually, if you look at the capsaicin receptor from birds, it is not activated by capsaicin. It's activated by heat, but it's not activated by capsaicin. And so that was another thing that kind of really captivated me, this idea that these really fundamental properties can be reflected in actually like very, very small, subtle changes in the sequence. I mean, I guess that's a theme that is probably also true in the odorant receptors, for example. There's a lot of examples of that in GPCR biology as well, but definitely something that was really, really interesting about that system.
I thought so too. I think it's very interesting that you have the same receptor in humans to detect heat, and also the capsaicin molecule to signal to the brain that actually the end result is the same. There is a burning sensation.
Exactly.
Comes through it.
Yeah.
I think it's just fantastic. And then the next question I had was about, actually it's more of a thought than a question. I loved how you got interested into the developmental biology, and you're right. I mean, going from the fusion of two cells to a full human, it's just a magnificent thing. And there are so many things happening at the same time that need to be perfectly orchestrated, coordinated. And I think it's just there's so many questions that need to be answered. But the question I had for you is, so you mentioned that you got interested in developmental biology, which led you to GPCRs somehow. But what was the first time where you actually heard about GPCRs?
Yeah, okay, that's a good point, because definitely like in graduate school, like in the Julius lab, even though most of the work going on in the lab was about ion channels, GPCRs were sort of always floating around there for various reasons. I mean, one reason was just that it was a sensory biology lab, and so we're really aware of sort of the odorant receptors. All of those are GPCRs, and some of the great work in that system. But also these ion channels, like the capsaicin receptor, they're actually modulated by GPCR activity. And this is a really critical way that they're sensitized, for example, in response to tissue damage. And so there's a lot of examples of that, actually, of GPCRs, mainly acting through GQ coupled receptors on PLC to sensitize these. They're called transient receptor potential channels, or TRP channels. And so it was one actually really nice story that came out of David's lab that was basically right before I got there, of GQ coupled GPCRs basically doing this and really powerfully sensitizing these receptors. But even though it was a GQ coupled pathway, it wasn't through diacylglycerol and PKC or IP3 the way that a lot of those pathways were. And it turned out that it was through PIP2, and it was actually PLC that was cleaving PIP2 to relieve this tonic inhibition of the channel. And I thought that was a really cool idea, and in part because I had learned a little bit about GPCRs already, and I knew that was not sort of the typical way that people thought about GQ coupled pathways. And I think you never really know as a scientist kind of how all these experiences that you have are going to eventually like influence your later work, but I do think that probably did have some influence on me in terms of thinking that just because a GPCR pathway is supposed to work a certain way doesn't necessarily mean that it really does work that way. And a lot of times you have to think about whether or not the ways that these proteins communicate with each other are really the way that you learned about, or is it potentially something different? So I think that was the first time that I really learned that was probably the first experience I had learning about GPCRs. But I really started to dive into them a lot more when I was a postdoc. We can get into kind of the reasons for that in a little bit. But basically, I sort of recognized part of the way through my postdoc that there was this sort of that the GPCR field and a lot of the tools and the approaches were going to be really, really useful for understanding the biology around surrounding Smoothened.
It's great. Actually, that was going to be my next comment / question, because you and I first chatted almost face to face at the Transatlantic ECI, and we were just talking about the fact that you've really dug deep into the tools used to study GPCR function and applied it to Smoothened and to the research that you're doing. But we're going to get to that in a couple of minutes. I asked this I asked this question to all my guests. Do you have a favorite GPCR, or maybe a favorite ion channel?
I think my favorite GPCR has to be Smoothened. I have to give it to Smoothened. And part of it, I guess, is that I think, the biology around it has been really, really interesting and rewarding to work on. I mean, it's been so cool to try to figure out some of these really basic questions about how Smoothened works. But I think part of the reason I have to say Smoothened is that I think it's also really taught me a lot of biology. Like I used to think, maybe kind of naively, that I had a pretty good understanding of how GPCRs get activated, broadly speaking, and how they signal downstream. And I think Smoothened has basically taught me that I don't understand that at all, and that there's actually a lot more to it than I ever realized. And so I think it's been kind of a humbling and a very educational experience, I think, to get to work with this receptor.
And you made a great point when you mentioned that, yes, we know so much about GPCRs, but at the same time, we don't know enough.
Yes.
And
yes.
As a scientist, not assuming that something is the way it is, but instead going at the idea or asking a question in order to explore how things happen for that specific system is key to any scientific work.
Definitely. Yeah, definitely.
So I think you're my first guest ever who works on Smoothened. So
I think that's probably true.
Tell us a little bit more.
So yeah, Smoothened is this, really, I would say it's really the key, the key signaling protein in the hedgehog pathway at the membrane. And so Smoothened has to, basically it has sort of two really critical jobs in the hedgehog pathway. So it has to be activated somehow. It has to go from being in an inactive state to an active state, when the hedgehog pathway is on, it has to be turned on. And once it becomes activated, it has to basically signal across the membrane to initiate sort of a pretty unclear series of events that eventually lead to activation of a transcription factor called GLI. And GLI is the major transcription factor in the hedgehog pathway, and that's really what's turning target genes on or off to, basically to ultimately to determine a cell's fate. And so, and Smoothened is also overactivated in a number of cancers as well. And so basal cell carcinoma of the skin is actually the most common human cancer. There's about 3 million, there's about 3 million cases every year just in the US. And I have to make sure I have that number right. But
it's okay.
Yeah.
We can amend it on the
a lot of cases. And it's almost always driven by overactivation of Smoothened, either through mutations in Smoothened itself, or more commonly through mutations in upstream pathway components that lead to sort of excessive activation of Smoothened. And then medulloblastoma is a very common pediatric brain tumor, which is also the same thing, driven by overactivation of Smoothened. And so Smoothened is also a major oncology target. And really important sort of basic science, also really important for therapeutics. But when I started my postdoc, we really had almost no idea about how Smoothened got activated or how Smoothened signals downstream. And so maybe I can try to put that into perspective a little bit for some of the listeners. So I think, for people that are working on GPCRs, right? I mean, they're all different from one another. But in most cases, if you're thinking about how a GPCR gets activated, usually you're thinking about a ligand. And either the ligand, it could be a peptide or a small molecule or another protein, but generally, that there is a ligand. You might know what the ligand is. You might not. It might be an orphan GPCR, but you can be pretty sure that it's going to be activated by some ligand. And then after it gets activated by a ligand, right, a GPCR is going to engage G proteins or in some cases, and also arrestins to signal downstream. So for Smoothened, we actually had no idea when I started what turns Smoothened on at all. Is it a ligand, or is it something altogether different? And part of the reason for that is that Smoothened is really unusual in the GPCR superfamily because its activity is being regulated by another membrane protein called PASHD. And so PASHD is this 12 transmembrane protein, and it blocks Smoothened activity. And that's actually what keeps the hedgehog pathway off. And when the hedgehog pathway gets turned on, so in a developing tissue, when hedgehog protein comes along and wants to activate the pathway in a cell, it binds to PASHD and inactivates PASHD. And then that, because it's a double negative and it's really easy to get confused, but because there's an inhibiting an inhibitor, that turns Smoothened on. And so that process of how PASHD communicates with Smoothened, I mean, nobody knew how it worked. Does it involve some kind of a ligand? Is it something altogether different? Okay, and then once Smoothened gets activated, it has to signal downstream to these GLI transcription factors. But it didn't seem to be something that used G proteins. Unlike pretty much every other GPCR pathway that people think about. And so, yeah, I mean, I think it's worth emphasizing this maybe for the podcast, because we really had almost no idea, almost like what planet we were on in terms of figuring this out. And it was something that I found to be, really challenging for sure, because there's not really a clear sort of concept that you can sort of plug into. It's not like you can just plug and chug and have a model that's worked for or an idea that's worked for other GPCRs and plug it in here, because clearly it doesn't work. But at the same time, it's also a really big thrill because you get the opportunity to work on something that's really fundamentally important. And making a discovery like this is something that is really something that I thought could be pretty impactful for thinking about development and about cancer.
That's so fascinating. I have a quick question though.
Yeah.
What is the topology of Smoothened compared to your rhodopsin or regular?
Right. Yeah. So it is a 7 transmembrane protein, and it definitely adopts the GPCR fold. And we have partial structures of Smoothened now, and definitely the membrane domain is very clearly in the GPCR superfamily, even if it is part of this outgroup. One of the unusual things about Smoothened is that it has a very large extracellular domain. And that extracellular domain is something that's unique to the class F GPCRs. And so just the Frizzleds and the Smoothens have that.
Yeah.
And in addition, Smoothened also has this fairly large intracellular domain that is also unique among GPCRs. And as far as I'm aware, it does not look at all like any intracellular domain from any other GPCR.
Wow, that's so great. It's funny because every time I talk to someone, or I go to a talk about GPCRs, I learned so much, but then I feel like there is so much to do, and it's so exciting.
Yes.
to learn about these new GPCRs. So you mentioned that actually in order to activate Smoothened, you needed the 12 transmembrane domain protein. That is it always co-expressed with Smoothened, or is it co-localized, or is there a shift to get those two partners together?
Yeah, so they're both PASH and Smoothened are generally expressed pretty broadly and at pretty low levels, but pretty broadly. And the other thing about this Smoothened that's pretty, I guess, interesting but also confusing is that it uses the primary cilium for signaling, and so Smoothened and PASH both localized to the primary cilium, this tiny little antenna-shaped structure. Although exactly why they have to do that, and what exactly is going on in the cilium, is really not that clear, even now. But they do seem to sort of be co-expressed and co-localized in that structure. So they do kind of get close to each other, although they don't interact physically. I mean, they don't, at least not in sort of conventional biochemical assays. They don't. It's not like a receptor and a co-receptor, for example. Okay. So we knew from that. I guess the field knew from that the signaling between them was going to be something pretty unusual. Didn't seem to follow that pattern.
That's so interesting. And what other proteins are expressed in the cilium?
Yeah, so the cilium ends up you can pretty much find all of the hedgehog pathway components in the cilium, and so PASH and Smoothened, of course, as the upstream players in the pathway, but also protein kinase A, which we'll probably get into in a little bit, a major, and GLI itself, the transcription factor, actually goes into the cilium. And then eventually comes out to turn on gene transcription. And so, yeah, we'll definitely, I'm sure we'll talk about that a little bit more later. But, one of the really critical downstream steps in the pathway involves PKA, which phosphorylates GLI and turns it off. And then Smoothened has to somehow be able to prevent that from happening. So you could see all the critical head shock pathway components in cilia, but also a number of other GPCRs are there. And so, it turns out to be this sort of little zone of GPCR signaling where you can find things like somatostatin receptors, some of the dopamine receptors localized there as well. There's some neuropeptide receptors. And so this is probably one of the reasons why if you have these mutations that affect the formation of cilia or that affect protein transport into cilia or out of cilia, they have this whole sort of spectrum of really kind of interesting and seemingly unrelated human diseases that involve, metabolism or neuromodulation or vision, and so they're called ciliopathies, but they all result from sort of mistakes in formation or maintenance of cilia. And so there's definitely some really interesting biology going on in there and some really interesting GPCR signaling. But, there's also plenty of GPCRs that don't use cilia. It's not like it's an obligate place in the cell for GPCR signaling. And so, this is stuff that's really being actively researched right now by a lot of labs. What is going on in this little antenna that it seems to be so important for these pathways to work.
It sounds like the little antenna is a very elite club of GPCRs and signaling molecules, and it also actually the localization must be so specific because of the function that these genes, these proteins have. And I guess the separation is the reason for the separation is obvious, is because it needs to be very specific, and you want to make sure that you activate only the correct receptors and the correct intracellular signaling pathways, which it's kind of a, I think all the events that happen in the cell are already so mysterious and so well coordinated and timed. And then you have the cilia, the cilia that have another compartment.
Right. And another idea there is also that there may be, so some of it may be compartmentalization, like you're alluding to, like you may want to have things that go on only in the cilium and not elsewhere in the cell to sort of compartmentalize them. But another idea is also that there may be factors in the cilium that are sort of unique to the cilium, like specific lipids or proteins or metabolites that are not found anywhere else in the cell. And some of these GPCRs may really need those in order to work properly. So that's sort of the answer may kind of be for different receptors and pathways. It may be some combination of those things.
Yeah. It's an elite club for whatever functions.
An elite club, but I guess to build on that analogy, a club where we don't exactly know what the membership rules are, because exactly. the receptors that have made it into that club, it seems to be a very odd assortment. But I think that's one of the fun things about working on it. There probably is some underlying logic to it that we just don't understand yet.
And my guess would be that it's very difficult to look at the signaling of the receptors and of the components of this little club separately from the rest of the cell.
Yes, that has been a huge problem, and actually we may want to get to that in a little bit when we talk about Smoothened. But one of the problems is that doing the cilium is really in a lot of ways still very difficult to access. You can do imaging on it, but it's quite complicated because it's so small. It's only a few microns long. It's only a fraction of a micron wide. So it actually has about the volume of a bacterium. And so, you can imagine how difficult that is. And you really can't do much biochemistry on it. There's not really a clear way, in the field to sort of, or a reproducible procedure for separating the cilium away from the cell body, as it's called. Yeah. So the biochemistry is very, very limited. And also the kinds of functional assays that you can do in the cilium are also really, really limited. And so I think, because you asked a little bit about how I got into studying GPCRs. So I think that was something that I really struggled with, I think, early on in my postdoc. And so to take a step back for a second, there are these huge mysteries surrounding Smoothened and how it works. But because the people that had worked on Smoothened in the past had mostly been cell and developmental biologists, there was really this idea that it's very important to study Smoothened in sort of the most physiological system possible, meaning in the cilium, endogenous protein or something expressed at very close to endogenous levels. And I think what I kind of realized after doing that for a little while is that it's incredibly difficult. And there's just a lot of limitations to what you can do in a system like that. And some of it really was coming back to this idea that like, oh, whatever is activating Smoothened has to be something special that's just in the cilium. And so you can't really study it in any other context. I mean, that was maybe my perception of it. But what I came to realize was that, if we don't figure out some better system to use to be able to manipulate Smoothened, it's going to be so difficult to solve these questions. And really in the cilium, the imaging was really difficult. The biochemistry was almost non-existent, I would say. And the kinds of functional assays that people could use were things that were like really, really far downstream of some of the things that you're probably used to looking at for GPCRs, like conformational readouts or membrane proximal sensors of activity. And that was really how I started to get really deeply into GPCRs, was that I realized that we have to have some better system than this, something that was sort of simpler, easier to manipulate. And I think this is something that people in the GPCR field probably just take for granted. Like, if you're studying a chemokine receptor, for example, like you don't have to study it in B cells. You can also just express the gene for the chemokine receptor in HEC293 cells. And yes, it doesn't recapitulate all the biology, but a lot of the key properties that actually does recapitulate. And you can do ligand binding, you can do G protein coupling, you can search for factors that regulate activity. You can look at all kinds of mutants. And so, a sort of a simplified system like that is something that's really powerful because it just opens up this whole new set of tools that you can use to study activity. And so, I started at that point to, I think, really dive deeply into GPCRs, because what I realized was that even if Smoothened is this oddball member of the GPCR family, and even if this idea about ligand binding or G protein coupling is not really correct for how Smoothened works in its canonical pathway, at least, at the very least, the kinds of like tools and the kinds of approaches and the kinds of thought of ideas in the GPCR field, I just had the sense that they were going to be relevant. And I guess most importantly, in a practical sense, there weren't a lot of people that were doing it that way. I mean, most of the field was really looking a lot more at these really in vivo systems and cilia. So I started to really read a lot about GPCRs. I started to go to a lot of GPCR meetings, and I would just sort of, I would kind of sit there and hear all these interesting talks and just kind of like write down, okay, here's like this technique I could use. Here's that technique I could use. And it was almost like it was like going to a shopping mall and kind of having all these things presented for me. But I also started to kind of go back into the GPCR literature too. And I read a lot of the papers from Bob Lefkowitz and from Brian Kodulka. And then even, farther back than that, I went and looked at some of the papers from like Martin Rodbell and Al Gilman on their work on G proteins. And I think what I kind of realized from all of that was that in all these cases, it was always some very simplified, accessible system that they used in order to make these big discoveries. Like one example that really inspired me was I read the paper from Jeff Benovic when he was in the Lefkowitz lab about the identification of beta adrenergic receptor kinase, which is now GRK2. And it was like a deceptively simple system. It was just beta adrenergic receptors purified, reconstituted into lipid vesicles. And then it was the start of this sort of massive biochemical fractionation effort to discover these kinases. But that ended up just being such a fundamental discovery. And it wasn't done with, endogenous receptors and kinases and so it was all done with biochemistry using a very simple, relatively straightforward readout, which is just phosphorylation of the receptor. And that was the kind of stuff that I really wanted to do with Smoothened, just to use these kinds of reductionist approaches and these simplified systems. I felt that that was the key to cracking all of these mysteries.
I love that you made so many good points. I'm going to try to take them apart. I love the idea. Yeah. So we agreed on the fact that Smoothened is a complicated system. People have been using, more physiologically relevant systems to study this, electrophysiology, but then we haven't gotten all the answers we wanted. And I love your approach of, taking it out of the context and then putting it in a very simple system. And the simpler is the better.
Right.
In order to understand
and this is, I think, a pretty prevalent idea in the GPCR world. I mean, I feel almost, it's something that we all take for granted so much that it's almost like not even worth saying it. This idea that you just express the receptor in a hex cell to start to learn about it. But, it's really interesting how when you go to a slightly different field, that idea is not as widespread, for example.
I love it. And I think you're right. And I think in the GPCR field in general, we got to a point where if you have a receptor of interest and you express it in hex cells and you do your regular quote unquote signaling assays, people are like, yeah, and so what?
Yeah.
But then coming at it from a different field where people don't even think about this, doing it this way, then it actually regains that value. And I think understanding how a GPCR works, any GPCR or any molecule, you have to put all these approaches together. The way I see them is little boxes.
Absolutely.
You express it in hex cells, you see what it does, you get some answers, then you put it back in a model organism or in a more physiological context, and then pulling the data together is what's going to give you the more information.
Yeah, it's sort of like the analogy with the elephant and how everybody's sort of touching a different part of the elephant and they all describe it differently. But exactly. I mean, basically, I think the thing that I always try to keep in mind is that every model system that you use in science has strengths and weaknesses. So there's no single model system that could ever answer all the questions about how an aspect of biology works. And so, the kinds of top-down approaches that people in developmental biology often use, which is to take an embryo and start to just do genetics on it, for example. That's top. I would consider that to be top-down. This kind of approach is more like bottom up. It's like, well, we have a critical component, and let's figure out what is the very simplest system that we can use to start to study its behavior. And you're absolutely right. Like all these things together are what ultimately is going to give us, a more complete understanding of how something works. But you have to, I think, recognize, when a certain kind of a model or kind of approach is not being used, that's something that you should try to go towards, because the chance of finding something new is going to be a lot higher there.
Especially with all the new biophysical tools that exist to study GPCR structure-function relationships. Let's get into that, because I'm curious. So what did you do with Smoothened, and how are you applying these biophysical methods to study and better understand the function of Smoothened?
Yeah. Okay. Yeah. So let's definitely get into that. So I told you when I started my postdoc, it was really not clear what turned Smoothened on. So Pash had to inhibit Smoothened, and then somehow that was keeping Smoothened off, and somehow that process had to kind of go away in order for Smoothened to be turned on. We didn't know what it was. The idea of Smoothened having the idea that was sort of floating around at the time was that Pash might be a transporter, and it might be transporting some endogenous Smoothened ligand, or transporting or rearranging it, or somehow depriving Smoothened of some ligand that it needed in order to be turned on. So that was sort of the idea. That idea actually came mostly from sequence comparisons. And so the Pash sequence looked a lot like the sequence of these bacterial transporters that were involved in transporting lipids into and out of bacteria. And in particular, there was a link between some Pash-related proteins in invertebrates and cholesterol. So there's a protein related to Pash called NPC1 that's involved in cholesterol exit from lysosomes, for example. And so the idea, and again, there was no really direct evidence to support this, but the kind of idea was that Pash might be some kind of a cholesterol or some kind of a lipid transporter, and that Smoothened might have a lipid ligand. And another thing that was known at the time was that hedgehog pathway activity was really, really sensitive to changes in cholesterol levels in cells. And so if you had, for example, mutations that affected cholesterol biosynthesis, or if you used pharmacological agents that extracted cholesterol from membranes, you would just completely shut down hedgehog signaling at the level of Smoothened. And so that had sort of led to these ideas about cholesterol being potentially like something that would change Smoothened's activity state. But it was still really, I guess, our understanding was still really limited because, a lot of these things, again, were happening in intact cells.
Yeah.
And, we didn't know, for example, if you depleted cholesterol from cells, I mean, those agents deplete every sterol from the membrane, right? And there's not just cholesterol, but there's hundreds of cholesterol metabolites around. And in addition to that, and so from that, we don't know how, from interpreting that, does that mean that Smoothened is actually being activated by cholesterol, or is it some other molecule, right? And then also cholesterol has all these different effects on membrane properties too, right? I mean, with rafts, and that was another idea that was floating around. And so basically at the time, with those tools, we kind of knew that we kind of thought that lipids and particular sterols were really critical, but there was not like a really clear idea of what they were doing. Are they binding to Smoothened directly? Are they having these physical properties? How important is cholesterol really the thing that's turning Smoothened on, or is it just facilitating the action of another ligand? And so I got to that point. We got to that point, and I just thought, what we need is in vitro reconstitution. This is something that is like really a very clean and a very simple way to address this question. So if we can purify Smoothened and put it into some kind of an in vitro system with a functional assay, then we can directly test, first of all, does cholesterol directly activate Smoothened versus something else? And, does cholesterol actually, does it potentially have a physical effect, or could it be binding directly? And so, from going to GPCR meetings and from reading work from Brian Kobilka and others in the field, they had made a lot of progress in two fronts. The first front was figuring out ways to express and purify biochemical quantities of GPCRs. And, so that had really taken off before I started my postdoc. And then the other thing was also the nanodiscs, which are, a really popular system in GPCR biology for biophysics and for structure. But the really cool thing about nanodiscs is that it's a fully defined in vitro system where you have complete control over everything that goes into that, including the composition of the lipids. And that was a system where, I could basically just reconstitute and reconstitute Smoothened and put in whatever lipids I wanted to, and just ask what the effect on activity was using a conformational readout.
Yeah.
And by doing that, we actually figured out that cholesterol alone is able to activate Smoothened to near maximal levels in that system. So again, this is really different from a lot of other GPCRs because, there's many examples of GPCRs that are regulated by cholesterol, like the serotonin receptor, for example, is regulated by cholesterol levels, but still its ligand is serotonin, right? I mean, serotonin is the main event there. Whereas for Smoothened, all you needed to put into these nanodiscs, besides simple phospholipids, was cholesterol, and then that turned it on. So that was an example of, I think, a really like a conceptually simple experiment where it really drew on, a lot of the tools and a lot of the ideas that it really had just been applied very nicely to other GPCRs. And it did take some time to figure out how to apply them to Smoothened, but really all that we did was build on some of the work that was already out there and just be able to bring it into our system. So
I love it. And did you tag first of all, was it easy to purify Smoothened? I think the challenge was with a lot of GPCRs in general, but with a lot of, exotic, let's call them exotic GPCRs, is purifying them. So one, were you able to purify it? Two, what was the readout like? What did you measure?
Okay, so the purification and the expression actually ended up being pretty straightforward. We end up using, we used SF9 cells a little bit, but we tend to rely more and more on mammalian cells and sort of the suspension mammalian cells and the BACMAM systems that have been really, really effective in producing a lot of GPCRs. It turns out that Smoothened actually works really, really well for that. And you have to make some truncations and some sequence modifications, but that's pretty standard for a lot of GPCRs. And so we did have to play around with that for a bit. But once we did that, it was actually fairly straightforward to be able to use those systems. So in terms of the readout, this is now we'll probably have to bring G protein coupling into the picture.
Mm-hmm.
And, at the time, so I will say that, Smoothened, it definitely was known before this that it can couple to G proteins in vitro. So if you expressed it in 293 cells, it would turn on, GI, GO coupled readouts. The thing that was really confusing to a lot of people was that, they had thought, the field had thought for a long time that because Smoothened is really good at coupling to G proteins, or to inhibitory G proteins in particular, that might be the way that it's signaled downstream. But the issue was that, if you block inhibitory G protein signaling with things like pertussis toxin, many labs had tried that experiment and had really never seen any clear effect on activation of GLI. So, for a while, the field, it depends on which field you're in. I think in the hedgehog field, a lot of people were sort of staying away from G protein coupling because it wasn't that clear if it was something physiological or not. But what I sort of thought at the time was, okay, so Smoothened can couple to G proteins. So why don't we just use them as basically like a proxy for whatever the real downstream readout is? Because it's coupling in a way that depends on Smoothened activity state. We did a lot of pharmacology on it to validate that all the same agonists and antagonists, synthetic agonists and antagonists that turn Smoothened on or off. They do in the GLI assays. They do the same thing in the G protein assays. And so I just thought, well, this is a great proxy because the thing is we don't know what's going on downstream of Smoothened, but we do know how to measure G protein coupling. There's a million ways to do that. And, G protein coupling is something that happens within a second, after most receptors get activated. And so I just did basically sort of the old school GTP gamma S binding assays in these nanodiscs, and it worked very, very well. And so that ended up being kind of our proxy readout for Smoothened activity state. But, to be fair, I think at the time, people, including us, were kind of wondering, the big question is, okay, you can do this stuff, but is this really reflecting the same molecular events that are happening when Smoothened signals to GLI? And by and large, I think the answer has been yes. It's not the conformation that couples to G proteins is probably not exactly the same as the one that couples to GLI, but the basic conclusions I think do hold. And so that I think ended up being a really helpful thing about being in the GPCR world, was that I became aware of sort of this, all the tools for measuring these kinds of things that I didn't know about before.
That's fantastic. I love it. One other question regarding the purified Smoothened. You mentioned that you had to modify it in order to be able to purify it. What was the extent of the modifications?
Not too much. One thing that we definitely had to do, so Smoothened has this very long unstructured C-terminal tail that actually is essential for communication to GLI, although it's not needed for G protein coupling, it turns out. So we had to lop that off because without that tail, appears to be unstructured in solution. We can actually get into that in a little bit later. But so we had to lop that off. And then there's sort of an unstructured domain at the very, very end terminus of the receptor that we got rid of. But we just replaced it with, basically used the same kind of system that the Gobilka lab and others used with flag at the end terminus. And it just turned out that we could plug into that really easily. And it worked really well. So that was really just the extent of the modification.
That's awesome. I really love it. Has anyone tried, or maybe you did try to, since you mentioned that, yes, there is G protein coupling. Have you have you tried to express the wild type Smoothened tagged with some kind of a fluorescent protein and look at, for example, FRET or BRET interactions between Smoothened and G proteins in hex cells?
Yeah, definitely. So we haven't done so much with we've done a lot with BRET. Not so much with G protein coupling, but there are definitely some labs that have. Nevin Lambert's lab has done that. And Gunnar Schulte also has done some of that. Yeah, we did experiments where we would express Smoothened in 293 cells, and then we would look directly at cyclic AMP formation in live cells, and we could definitely see really nice effects of Smoothened on those readouts. And we were pretty confident it was through G protein coupling because that could be blocked by things like pertussis toxin. Okay. So definitely, that was, again, one of the mysteries here is that, it's really good at coupling to G proteins, and yet that doesn't seem to be by itself, the full explanation for how it communicates with its physiological downstream target.
Yeah. So it seems to me that point of contact is missing. So basically, in order to be to do its function, so for smoothens to function in developmental biology, so to activate the transcription factor and initiate gene expression, the missing part is where does the receptor and the transcription factor interact?
Exactly. So that was really the $1 million question. And so, what we all wanted to figure out in the field was what is going on immediately downstream of Smoothened. If it's not G protein coupled, if it's not G proteins, what is it? And that was So we actually, I can talk about that in a little bit, but we actually made, we think some really critical progress on that recently, which has been really exciting because for a long time we're all just, I was scratching my head like, how on earth is this working if it's not through G proteins? But we think we have an answer for that. So.
And what is the answer for that? If you can say.
Oh, of course. Yeah. So this is all published. So we put out a paper recently in PLOS Biology. It just came out in April from my lab. And then we have also a new preprint that we just put up in the beginning of July that kind of talks about kind of the next step in this. But the short answer is that what Smoothened does is it has a way to block PKA signaling, but not using G protein. So it's very unusual, or it seems to be very unusual. But what Smoothened does is that it turns out it directly binds to the catalytic subunit of protein kinase A, which is very, very unusual for a GPCR. And so PKA, as I pointed out before, is this incredibly important regulator of GLI. So PKA phosphorylates GLI, and that turns it into an inactive transcription factor. And so the idea was really that something has to prevent PKA from phosphorylating GLI in order to turn GLI on. And maybe Smoothened could be the thing that does it. But how does it work if it's not through inhibitory G proteins? So it turns out the answer is that it directly binds to the PKA catalytic subunit. And it does two things. So the first thing that it does is that it recruits the PKA catalytic subunit to the membrane, to the ciliary membrane, from the sort of, I guess, the rest of the cilium. And we think what that does is that it sort of sequesters PKA away from GLI, which is a soluble protein, and helps to prevent phosphorylation. So that was sort of that was really the subject of the first paper that we wrote. But then it turned out that was not the whole story. And the second part of it, I think, ended up being even more, surprising, which was that basically Smoothened has this decoy substrate motif in it that physically blocks the active site of PKA. And so it just basically wedges in there and just interrupts catalysis. And so it's, as far as I know, the only example of a GPCR that physically blocks the activity of PKA. And so that, ended up actually being, it was definitely not something that we were expecting to find, but it really explains how Smoothened can block PKA, but without the need to use inhibitory G protein. So it actually makes a lot of the experiments sort of make sense in the field.
That's so fascinating, and especially with a long C-terminal tail, you would assume that it has to interact physically with other proteins.
Right. That was the idea. I mean, basically, we all, the whole field, I think, really felt that Smoothened has to be interacting with something in order to signal to GLI. And so I can maybe tell you the story a little bit behind how we made this discovery. It was actually there were sort of two, really wild stories here that allowed us to do this. So the first was, I guess, identifying this connection between Smoothened and PKA. And so basically what happened was that, my lab is a pretty new lab, right? We're pretty small. And so I had a couple of undergraduates who came and wanted to work in the lab. And one of the undergraduates, Corvin Arbiseth, he recently graduated. But at the time, he came to my lab and I wanted to just give him a project to work on. And we were really interested in this question of Smoothened, downstream coupling, but it just seemed like, my gosh, that's like way too hard of a question to give to an undergraduate who's never even held a pipette before. And so, at the time, right, we were thinking about, okay, like you said, Smoothened has the C-terminal tail. It probably interacts with something. And we had the idea that maybe we would use mass spec to figure out what it was. And I thought, okay, maybe it must be some unknown factor. So let's try to use mass spec to figure it out.
Yeah.
And even that I felt was probably a lot to ask for somebody that had just, just come to the lab. And so I thought, well, why don't we start with something simpler? And so I just said, okay, well, before we go on this mass spec expedition, why don't you just test all the known components of the pathway to see if they interact with Smoothened? And I thought, it's probably not going to work. He'll probably get some negative data, but at the same time, he'll learn about how to work in the lab. We'll be able to write like a little undergraduate research grant about it, and he can get some funding. And so Corbin sort of went around and he went in and he sort of dutifully, cloned all, and I guess he cloned all of these genes into Brett vectors. And I think that was the one thing. I don't want to call it intelligent because I think that makes it sound like we knew what we were doing. But the one sort of smart thing that we did here was that we used Brett for looking at these interactions instead of things like co-IPs or co-localization studies, like a lot of other people in the field. And as you know, right, Brett is such a powerful technique. It's a way to look at protein-protein interactions in cells and really stringently assess things like specificity by doing saturation experiments and things like that. So Corbin, cloned all of these pathway components into Brett acceptor vectors, and he made a Smoothened Brett donor. And he went through, and he just discovered this enormous Brett interaction between Smoothened and the catalytic subunit of pKa. And I remember very vividly when he told me about it, because I was actually at a conference. It was a Cilia conference, and the night before I had stayed up kind of late with a friend of mine, a colleague, and we were talking about some of these questions at Smoothened, and we were kind of lamenting the fact that, like, is anybody ever going to figure any of this out? It seemed like such a murky problem, and I kind of went to bed that night after that conversation thinking, like, oh gosh, are we ever going to figure this out? And then the next morning, as I was getting ready to leave the conference, then I saw Corbin's data, and I saw this huge Brett signal, and I was like, oh my gosh. And I didn't know what was going on at the time, but I thought this has got to be something important, because the major positive regulator in the upstream part of the pathway, the major negative regulator, they're talking to each other somehow, yep. Right? So that was sort of the first, I guess, story behind this. And so it ended up just, even though this was just one person in the lab who sort of did this, I guess in retrospect, it was sort of a bold experiment, even though I think it was actually kind of a naive experiment on my part, but it ended up leading to this discovery, and it really kind of sucked everybody in the lab into it. And we spent the next year trying to figure this out. And so that the result of all that work was the PLOS biology paper. And the thing about that paper was that we knew that Smoothened interacted with pKa. And we also knew that it depended on Smoothend's activity state, because it turns out that GRK kinases, right, mm-hmm. They're actually moonlighting in the hedgehog pathway. They recognize the active state of Smoothened and phosphorylate it. Mm-hmm. But instead of triggering arrestin binding, like it does for most GPCRs, it triggers pKa catalytic subunit binding. So yeah. So and it actually makes a lot of sense, because it's known that GRK2 and 3 are very strongly required for hedgehog signaling. So we had this idea, this model, right? Okay, Smoothened sequesters pKa at the membrane, and that's how it prevents glee from being phosphorylated. Yeah.
I was just going to say, I love the simplicity of the experiment. I love the fact that you just, just going through the exercise and trying to look for a protein-protein interaction with a simple live cell method.
Yes.
And, yes, you're right. Maybe it would have been nothing, but I think for two things, for a student, for an undergrad to go through the exercise and really be systematic. I think that's a great experience.
Absolutely.
But then to be so lucky to actually detect something really cool.
Yes.
And then building a whole paper around it, and it's an important discovery. I think it's just it's the awesomeness is twofold at this point.
Oh yeah, and I think the other thing I learned from this is also that, and I think this is maybe something worth emphasizing for the audience here, right? Is that a lot of young scientists sort of assume that you need a lot of training and a lot of experience to make a discovery, but that's actually not true. And I think scientific discoveries are things that, in principle, any of us can make. Whether you're a Nobel laureate or somebody just starting in the lab, really what it comes down to is, working hard, being willing to try something new, and also being lucky. But
Yes.
it's very kind of democratizing in a lot of ways, because a discovery is something that really is within reach for everybody.
I think so too, and I think the fact that reading up in the literature and trying to think outside the box, or taking that 3,000 feet overview of a question allows you to ask, or not to ask simple questions which have a deep impact, but also
Yes.
allows you to design very simple methods or very simple experiments and direct experiments to answer that particular question.
I totally agree, and I think that I feel like in biology, right, finding the right tool for the job is a big part of how you do science. And I think for every question, you got the right model system and the right approach, right?
Yeah.
And I think in this case, right, Brett ended up being exactly the right approach to use, because it's a way to look at protein-protein interactions in a living system, without having to do a lot of biochemistry and in vitro reconstitution, which ends up, we are doing that now for this interaction, but it ends up being pretty tricky. And I think if we had started with that approach, we would have almost certainly missed this. So, some of it was maybe I can't say that I had a lot of foresight about it, but I think from going to enough GPCR meetings and reading enough papers, I did know that Brett was such a powerful technique, and I did feel like it had a lot of potential here, because people weren't really using it so much to study this receptor.
This is awesome. So before we move on to the next part of the next paper that you mentioned that came out in July, I want to take a moment to talk about the ligand. So you mentioned that you used Brett to determine this interaction, to discover this interaction between the C-tail of Smoothened and the active site of PKA. Right. What type of ligands did you use?
Yeah, so one thing that was good for Smoothened compared to other class F GPCRs is that there's a lot of pharmacology for it, and so we have really good, endogenous, synthetic and naturally occurring also, agonists and inverse agonists. So if you just express Smoothened in 293 cells, for example, or any cell for that matter, as long as you don't have PASH around, or if PASH is at low levels, Smoothened is going to be constitutively active, because it's essentially going to be binding to the cholesterol in the membrane. Mm-hmm. And just as a side note, we actually, when I started my lab, we actually had a really nice collaboration with Ashish Manglik's lab at UCSF that's still ongoing, where we helped to determine the active state structure of Smoothened, stabilized by a nanobody. And we actually figured out, from that exactly how cholesterol binds and how it triggers activation. And it turns out that, so it's sort of allowing those experiments I started as a postdoc to really come full circle, because we realized that cholesterol directly binds to Smoothened and triggers the same activating conformational change that it does for other GPCRs. So just expressing Smoothened in 293 cells, for example, in these Brett assays will lead to constitutive activity as long as PASH is not co-expressed, for example. But we do have positive allosteric modulators. There's one that's called SAG, Smoothened agonist, that will activate it to even higher levels. And we have really good inverse agonists based on cyclopamine, which is a compound that actually has a really interesting history, especially related to Utah. But basically, it's a teratogen. It produces birth defects, and that was how it was discovered. But it turns out to be a really powerful inverse agonist for Smoothened. So we have really good ways to turn Smoothened on and off in these assays, which is, I think, one of the blessings of working on this receptor, is that there's good pharmacology.
That's amazing. Where does cholesterol bind?
Yeah, good question. So it binds in a couple of locations. So from our structure, we figured out that the major cholesterol binding site on Smoothened is within the transmembrane domain of Smoothened. And so Smoothened is a little bit different from other GPCRs. It has a particularly long, binding pocket that's pretty large in volume. And so cholesterol turns out to bind really in the deep part of that pocket. And in doing that, what it does is it actually triggers a conformational change in the receptor that's, again, very similar to the activating conformational change in other GPCRs. So that's one place that it binds. Another place that it binds is also in the extracellular domain of Smoothened. And this was something that was discovered even before our structure. It was discovered by a number of other labs. And so there's a cholesterol binding site there as well. And then in our structure, we used SAG to stabilize the active state, which binds in the membrane domain, but just a little bit north of where the cholesterol binds. And now some recent work has shown that cholesterol can probably bind in that part of Smoothened as well. So it turns out that there's multiple cholesterol binding sites in Smoothened, but all of them work together to help stabilize this active state of the receptor. And the allostery there, is incredibly interesting and incredibly complicated. And is something that we definitely, want. It's really worth figuring that out. How that works.
Yeah, actually, my next question would have been, how many cholesterols does it take to activate Smoothened? It could be.
It sounds like the start of a joke. Yeah, it's like how Smoothened walks into a bar, and yeah.
And yes.
Yeah, so that is a good question. So it's not clear, and I think I could definitely imagine that under some circumstances you might have one or two of these sites occupied. And under other circumstances, you might have all the sites occupied. And I think, coming back to the biology of the system, that might be a really important way that nature sort of like creates this system where you kind of stabilize discrete activity states of Smoothened.
Yeah.
Because remember, in development, right, the amount of hedgehog protein, which is really what Smoothened is reading out here through the activity of PAST, right?
Yeah.
Those amounts are really, really important. That's what's going to determine, for example, what kind of a cell, kind of a fate a particular cell adopts. And when you have these proteins that are in gradients, right, like hedgehog is, a very small change in the amount of hedgehog actually has a pretty big effect on what kind of a fate a cell can adopt. And so Smoothened has to be able to measure these things really precisely. And so I think the idea of, this is a little bit of a maybe a naive idea right now because we still don't understand all the allostery. But the idea that you would be able to stabilize like sort of a low activity state or a medium activity state or a high activity state, could be a way to kind of allow Smoothened to accomplish this graded signaling that it needs to do in order to pattern tissues and organs.
I love it. I can. We were talking and I could see that the fact that the amount of hedgehog interacting with its patch would determine how many cholesterol molecules can bind to the receptor, which would then depend on how many pKa's
exactly
would be inhibited.
Exactly.
I think it's just phenomenal.
Yeah, it's been such a fun system to work on, and I think again, taking a step back, right? Like there's so many examples of cholesterol, regulating GPCR activity, but I think this is probably the most direct example where cholesterol really is the ligand, and it really is also the messenger in this pathway, right, between PASH and Smoothened, that actually accesses the binding site through this tunnel in the bottom, and so playing a role like this, it really is playing the role of a ligand in this for this receptor, and so it's just this very direct. It's really the main event here. It's not a supporting actor in any sense of the imagination.
Exactly. I love it.
Yeah.
I really love it. So you were mentioning that you recently published a new paper.
Yes. Yeah. I would definitely talk about that. So that's still in the preprint stage right now. And so we just put it up in the beginning of July, but that ended up being another really wild story. And that was coming from this model about, okay, we knew Smoothened and pKa interact, but to go from that to Smoothened actually inhibits pKa activity. And so the way that this unfolded was just really kind of a wild ride. So, we started to write this PLOS biology paper during the pandemic. And, I was sort of stuck at home. My lab members were all sort of sequestered at home, and we had a lot of fun writing the paper together. And so we wrote this paper, and I just felt like, pKa has been studied for decades and decades, and we kind of came into it sideways like this, right? And I just felt like, I really should show this to an expert in the pKa field and just get their input on it, to know that we're on the right track here. And so I just sort of sent a cold email to Susan Taylor at UCSD, who's really kind of one of the world experts in sort of the biophysics and the structure of pKa. And she, her lab had made all these seminal contributions to pKa. Structure over the year, the first structure of pKa, the first structure of a kinase ever, actually.
Wow.
And so, I sent her an email, and and I told her a little bit about our work, and so we quickly set up a meeting, and she was also stuck at home, because the pandemic was affecting all of us. And so, I kind of wonder in retrospect whether any of this would have happened if it hadn't been for the pandemic. So it's maybe one silver lining. So anyway, we showed her, I told her about our story, and she's like, oh, this sounds pretty interesting. And then she just said, well, you think PKA is binding to the C-tail, like, you know anything about that? And I said, well, not really. So she said, well, let me see the sequence of the C-tail. And so I showed it to her, just the figure from the paper. And within about 15 seconds, she had recognized, oh my God, there is a sequence here in the Smoothened tail that looks like a PKA inhibitor. And so this just totally blew my mind. And just to give a little bit of background here, typically the things in cells that regulate PKA activity are what are known as pseudosubstrates. And so they will bind to the active site of the kinase, but just sort of take up space. And they prevent, just sit there, and they prevent the kinase from phosphorylating anything and from continuing catalysis. So they're really like decoy substrates. And so this protein kinase inhibitor, or PKI, is really like the classic example of that. And that was, something that Susan was very, very familiar with, because that was a sequence that she really knows by heart. And so she quickly picked out this sequence, and then it just became very, very clear to both of us, like, oh my gosh, Smoothened is going to Smoothened is going to stick this thing directly into the active site of the kinase and shut down activity. And that's, how it's going to, that's going to be the other critical part of this mechanism. And so that just basically launched this collaboration over the last, 8 to 10 months or so, involving both of our labs and a number of other labs too. And it's just been so much fun, because I think, we didn't really know anything about PKA starting this, and they didn't know anything about Smoothened or Sonic Hedgehog, but we really brought different things to the table. And as a result of our collaboration, we now, I think, have a really clear idea that Smoothened does use this decoy substrate motif to directly block the activity of PKA. And so it just really ended up being, a lot of fun for all of us.
How cool is that? So, a little sequence getting so many people involved in a great project. And you wouldn't think that, yes, PKA function is important, but as someone who's never really worked directly on PKA, in my mind, okay, it phosphorylates stuff and it does whatever it needs to be doing, but really connecting it to something so important as Smoothened function, I think is just, it takes it to the next level.
Yeah, it was definitely not something that we expected to find coming into this, but it actually made so much sense, because we knew that this intracellular domain of Smoothened was critical, and I always felt in my heart that was the key to answering this question, is that we know it's critical for communication to glee, but what is it? What does it look like? Yeah. I can tell you, I spent many afternoons as a postdoc blasting it against everything I could think of, and coming up with nothing. And so you can imagine how much it must have, it really blew my mind the way that somebody would just look at this and see it. And it is hard to see if you're not looking for it, but all the critical residues are there. And so it was, I think it was also a lesson about how the human mind is really more powerful than blast when it comes down to it.
Yes. I think when you're an expert in your field or in your proteins, then you just look at the sequence and you just recognize, because your brain is looking for it already.
Absolutely.
Which I find fantastic. Yeah. I have two, actually more questions, but two and with regards to Smoothened. Have you ever done any experiments where you started really chopping off portions of the N-terminus or portions of the C-terminus to see how what was the contribution of the C-terminal and N-terminal tails of Smoothened?
Yeah, we did quite a bit of that. And so the N-terminus of Smoothened, if you chop it off, it turns out Smoothened is still able to signal downstream to glee, although it does have some problems. It ends up having sort of too much basal activity, but at the same time, no stimulation by hedgehog. And so that sort of puzzled us for a while. But we think the answer to that question is that probably this site in the membrane domain is really fundamental. But because there's also this site in the extracellular domain, and there may even be some possibility that cholesterol could even travel between them, that chopping it off is going to really get rid of a lot of that important allostery that's critical to get proper Smoothened regulation. So we did that with the N-terminus before. With the C-terminus, basically, if you chop it off, Smoothened can't signal to glee anymore at all. And it turned out that actually the other funny thing about this inhibitor motif was that we knew from other work in the field, if you chop off, if you make C-terminal truncations of Smoothened, basically the farthest that you can go is right where this PKI motif is. And if you go any farther, it doesn't work anymore. But again, we had no idea what it was or what it looked like. And of course, it made perfect sense once we knew what we were dealing with. And so, that was kind of our story with that.
That's fantastic. And then our next question, so you mentioned that there is great pharmacology out there to study the function on the activation or the inhibition of Smoothened, and expressing it endogenously in HEX cells because of the cholesterol yields a constitutively active receptor that can be further activated by agonists.
Right.
And taking this information and putting it in the context of various cancers, are there any drugs that target Smoothened in these cancers, or is there a potential of screening for drugs that
so
target?
Yes, that is a good question. So the answer is yes. There's actually Smoothened is quite druggable, and there's already 2 FDA approved Smoothened inhibitors that are used in a number of different cancers. And so it's actually a really excellent drug target in that sense. But one of the interesting things is that actually those drugs were discovered before we knew anything about what activated Smoothened. And so it turns out from our structure with Ashish, what we learned was that those drugs actually act as competitive antagonists for the cholesterol. And so that's how they work. But it turns out that they actually don't do that very well. They tend to bind more in the upper part of the pocket and extend a functional group downward towards the cholesterol. And so they will kick the cholesterol off. But one of the problems is that it becomes very, very easy to get resistance mutations in the clinic because, since there's so little overlap between the cholesterol and the inhibitor, it's very easy for the tumor to come up with a mutation in Smoothened that will block the binding of the inhibitor, but spare the binding of the cholesterol that's needed to keep Smoothened active. And so I think the challenge right now is dealing with those resistance issues because they come up very, very quickly and they really compromise the long-term efficacy of using Smoothened inhibitors to treat cancer. And so one of the ideas that we have right now based on the structure is that if we had an inhibitor that binds in the deep part of the pocket that really overlaps extensively with the cholesterol, that's probably going to be a much better inhibitor because it's going to be really difficult for the tumor to come up with a way to mutate Smoothened that would block the inhibitor without also affecting the cholesterol. And so that's something that we're actually working on right now. And I like that because I think it's a really clear example of the way that, you have to do this basic science investigation of how these proteins work to be able to come up with that. I mean, there's no way to come up with it otherwise. And so, definitely like that is an example of, some newer stuff that's going on in the field in terms of Smoothened therapeutics.
I love it. And what kind of, so as you mentioned, ideally you'd like a molecule that binds into the deep pocket, the deep region of the receptor to get rid of the cholesterol or to make sure, to stop the constitutive activity of Smoothened. What are some of the tools that you would be using to, for example, screen for such molecules?
Yeah, so certainly one could imagine doing things like structure, based drug discovery, in silico docking, things like that. It turns out, in another kind of funny coincidence here, there's already a real, a very, very potent and efficacious Smoothened inhibitor that binds in the deep pocket and actually overlaps extensively with cholesterol. And it's a molecule called SANT1, and it's a laboratory tool compound. And there was a structure, of Smoothened bound to SANT1 many years before ours. And the only thing people realized about it was, isn't it unusual that it binds in the deep part of this pocket? All the other inhibitors bind in the upper part. But now that we have the structure of Smoothened bound to cholesterol, now we know that actually that's desirable from this standpoint, right? And so it turns out we already have, a nanomolar affinity tool compound that binds in the deep pocket, and we're starting to use it now, in some of our experiments. And so it's, again, another funny example of how, a lot of the previous pharmacology in the field was really great and kind of came up with a lot of these inhibitors and kind of gave us a head start here.
That's fantastic. That's great. So that tool compound could potentially become more than a tool compound.
Yeah.
If it can be modified to hopefully get into the clinic or at least used as a template.
Right. And even if it doesn't, I think just the idea of like drugging the deep pocket, if it ends up being sort of a tool compound to establish that concept, would be really, really useful and would stimulate, could even, for example, stimulate, biotech and pharma companies to really mount a campaign to look for good deep pocket inhibitors for Smoothened.
What would be the best type of readout or signaling assay that you'd use to look for these type of molecules?
Yeah, that's a good question. I think you wouldn't want to do conventional drug screening because a lot of companies have done that. That was what led to the existing Smoothened inhibitors. Yeah. So it's actually pretty easy to find those. But again, they're not going to be drugging the right part of the pocket for the most part. Yeah. They almost all bind in the upper part of the pocket. Yeah. So I think probably, maybe something that, involved sort of a purified receptor system could be, a better way to look at that, or to maybe develop an assay with the deep pocket inhibitor where that was, labeled, and then one could look for compounds that displace that. Could be another way to potentially look at it. So I think the key is basically to do it not in an unbiased way, but in a way that sort of biases the system towards identifying these specific inhibitors that act in the specific way.
I love it. This is awesome. This is so exciting. So I guess I'll ask it because I asked this from everyone, but I think we've kind of went around and answered the question. Do you think GPCRs are still a good drug target?
Oh, definitely. Yeah. I mean, clearly they've evolved to be druggable, right? I mean, they've evolved to bind small molecules and other ligands with high affinity, and so they're really maybe one of the premier examples of a druggable molecule. And actually on that, so in that sense, they really are probably one of the best examples of, I think, a druggable superfamily. Yeah,
I think so too. So now that we're heading, let's take a step out of science and let's go back. I think we've kind of alluded to this throughout the entire discussion, where we talked about science, about your journey, but about the fact that you've always looked outside of the conventional toolbox and asked questions that were a little bit different and tried to apply one type of toolbox to a new question. What would be your advice to junior scientists who aspire to contribute to the field or to science in general?
Yeah, that's a good question. So I think the first piece of advice I would give is don't be afraid to ask a big question. I think a lot of trainees, again, sort of feel that like in order to make a fundamental discovery, you have to have like years and years of experience in science, and that maybe doing things like that is something that like a beginning graduate student, or even an undergraduate, is just out of their realm. But like we talked about before, I think, asking a big question, making an important discovery is something that really is within reach for any of us in principle, right? If you're asking the right question, right? And so I think that's really critical. And part of doing that as a young scientist is that you need a mentor to really help to guide you to like, how do you identify a good question and how do you ask it? And so I think it is really important, to find, a mentor and to find a lab that is doing that kind of science. So I think that's the first thing, is to ask an important question. I think the second thing is that I think it's also important to try to be different and to try to not do exactly the same thing that everybody around you is doing. And, I think part of it is that, as a trainee, I think you have to, you shouldn't just be reading, the papers in your field and going to the talks that everybody else is going to. I think you have to try to think, more broadly than that. And so going to, looking at what's going on in other fields, going to seminars for people that are not studying your specific question is really critical. And I think in our case, or in my case, I guess, because by really diving into the GPCR, world, right, which is something that people in my field weren't doing so much. I mean, it gave me all these new tools and ideas that I could run with. And so I think, finding your niche like that is really, really important to do. And I think, especially for people that are thinking about, like, if they're postdocs potentially looking to establish their own labs, having a way that you're doing something that's different from everything else that's going on is something that will really help with that process, because it kind of helps to explain to people like what is your lab and how does it fit into things, and that you're not necessarily just doing the same thing that many other labs are doing. And then I think the last piece of advice is to really try to think. It's really important to think long term and remember that science is very much a long game. So as a postdoc, I kind of recognized that I needed to establish some new tools and approaches to study these questions that interested me. And that it took a while, right? I mean, it took a while to kind of get all these ideas and all these tools in place. I think there's a lot of emphasis now on trying to be really fast and like really efficient with your training. And so, the K99 grant for postdocs is one example of that. And, the intentions for those kinds of things are really good. I mean, they're in the right place because I think that people should be able to become independent, sooner rather than later. But I worry a little bit about those kinds of things, because I think that they encourage people to think in a very short-term way. You have to kind of have a paper within the first, couple of years of your postdoc to be competitive for those grants. And so in a lot of ways, then that's already going to start to influence your thinking about what kinds of projects you pick up and what kinds of things you do. And I have to say that I think, I definitely did not set any land speed records during my postdoc by any means. But right now, I think our lab is really kind of reaping the benefits of a lot of the time that we put in earlier on this stuff, because I was in in labs that were really supportive of me taking time to do these things. So I think it's really important to think long term and to think about kind of where you want your research to be, not just like a year from now, but like several years down the road.
I love that, and I think there is a double-edged sword there when you talk about speed and getting data as soon as possible, and then trying to really take your time and explore and become an expert at your own topic. You mentioned your undergraduate student who joined the lab, and you said that he didn't know how to pipette to begin with, and I think that's we have to go through that. We have to go through that training where we get to a point where you can do the experiments with like that, and then you have that mental space to ask more broad and scientific questions and important questions.
Yeah, not everything in science is about efficiency, I think. And that was definitely something that I learned from being in David's lab and Phil's lab, for example, is that, they knew that, making an impactful discovery is not necessarily something that you're going to be able to do in a short amount of time. You have to, as you said, you have to spend some time exploring, and in a lot of ways, like figuring out what planet you're on, in order to be able to figure out like what is really at the heart of this problem and how do I get it. It takes a long time. It's not something that you can just sort of do very, very quickly. So
it's a marathon. It's definitely a marathon.
Absolutely, yeah.
But I think one key component and one key important thing that you mentioned is really taking the time to talk to other scientists, to bounce off ideas. And I know from experience that being an undergrad or a graduate student, or even a postdoc, sometimes you're just afraid to say out loud what your ideas are, but you shouldn't.
That's absolutely true. And I think another thing is, I think in order to come up with a good idea, you actually have to go through like a very large number of bad ideas, right? And just to even be able to recognize like, oh, that's something that has potential, versus, oh, that's something that's probably not going to work. I mean, you have to like go through that. And so I definitely came up with lots and lots and lots of kind of wacky ideas that we fortunately discarded along the way. But that is very much a part of the process.
I don't know if you've heard Bob Lefkowitz's episode, and I think he was mentioning that every year at the end of the year between Christmas and New Year's, he sits in his office and he makes a list of all of the things he wanted to do. And then if he gets through 20% of those things, I can't remember if it was 20% of the list or the success rate would be 20%, and he'd be very satisfied with that success rate. So.
Yeah, I don't think, but I think definitely in my postdoc, I don't think I was getting close to 20%. I think it was quite a bit lower than that. Which was a little scary at some points, but I totally agree with that. I think that is really important to that people know that when you're doing stuff that's out of the box, especially, it's going to take time, and you just have to expect that like there's going to be a low return phase in it, and there's going to be a lot of a period of time where things aren't working, and I really feel like that. to me is by far the hardest part of being a scientist, is knowing how to get through those periods. And so you can get to the point where you finally make a discovery, because you have no idea how long that low return phase is going to be. It could be a month, it could be a year, it could be 10 years. But the nice thing about being a scientist, though, is that it isn't just about raw brain power. It's also about experience. It's a craft as much as it is a sort of an intellectual discipline. And so with more experience, right, you get to know what the pace of a project is like, and then you get to, I think with more experience, I think I've learned when I'm really making progress on something, even if it doesn't look like I am, versus when I'm spinning my wheels. So that's all the part of this that I think comes with more experience.
I think so too, and it's an art as well.
Absolutely.
It's some way we're all artists at our own craft, our own levels, and I think you get more experience and you might get more confidence by doing it over time.
That is absolutely true. Absolutely.
All right, which gets me to my next question. Speaking of art and experience and confidence, what would be the top 3, or even less or more, aha moments that you had as a scientist that shaped your trajectory? I think we touched upon it a little bit, but let's recap.
Did, just to recap, I think I think the first discovery was probably the discovery about cholesterol activating smoothened in vitro, and the way that I think really told me a lot about how pivotal cholesterol was in smoothened activation. I think that was definitely an aha moment, and maybe along with that was also the first inkling with Ashisha's lab of how cholesterol really bound, and I actually remember that as well, because I had gone. I remember I was in the Bay Area, and I had gone down to the Bay Area for the day, and I hadn't brought my phone with me. And then when I got back home, this is basically my phone had totally blown up, because we had we made she had figured out that the cholesterol was in the structure and where it bound, and it was really exciting. So that was definitely the big aha moment, was I guess the combination of knowing that cholesterol was critical and figuring out how it worked. I mean, that was one thing. I think another aha moment was this first discovery that pKa catalytic subunit bound directly to smoothened. I mean, that totally blew my mind. And that was also super exciting. We talked about that a bit. And then I think lastly was this idea that smoothened has got this decoy inhibitor motif embedded in its tail that it uses to physically block pKa, and how wild that was. I mean, that was a huge aha moment for me as well. So I think those are the top 3. And I think one thing to add to that is that, and maybe this is also useful for some of the trainees in the audience, or maybe also for the people that are going to become PIs. You don't actually have to be a PI, do the work yourself to have that aha moment. And so I think that's one of the most rewarding things for me as a PI that I didn't realize starting this, was that if one of your trainees makes a big discovery, it's actually, you feel some of that too. Even though you weren't doing the pipetting and you weren't the person that really saw it for the first time, the fact that you're involved with it, it really gives almost that same dopamine hit that you get from making a discovery. And so that's actually been a lot of fun, for me to learn as a PI.
I think that's even more potentiated because, it's your student or your undergraduate or graduate student, and
absolutely,
you've created that environment for that person, for that budding scientist, to make that discovery. So I think the dopamine hit is double. As one for the scientific discovery, the second would be for you actually being responsible for another scientist's career.
Yeah, I totally agree with that. And I think that was something that I definitely didn't realize as a trainee, but the sort of excitement that comes from making a discovery is actually something that you could sort of divide up among many different people involved. And it's still just almost as exciting, I think, as if you did all of it yourself.
Agreed. All right, last question, and I ask this from everyone. Again, we have these go-to questions. Where can people find you? And if you have job openings, where can people reach out to you or find these?
Yeah, so we have a website, myerslab.org. We also have a Twitter account that I try to keep updated pretty regularly. And we are recruiting people right now. And actually, I would love to bring in some more people in my lab, especially people that have GPCR backgrounds, because I think a lot of the thinking and a lot of the approaches, from the GPCR world are really the kinds of things that we're doing in the lab right now. And so I think, people that are maybe finishing up, their graduate work and that want to be able to take some of what they've learned about GPCRs, but take it in a new direction and to think about, how does it influence tissue and organ development and how does it influence cancer? We would love to hear from people like that. And so, we have a job opening posted right now on my Twitter account and also on Job Archive. And I think I sent it to GPCR, Dr. GPCR last night. So maybe it will get up at some point as well.
Yes, it will. We'll do our best.
Absolutely. Yeah. So we would definitely love to hear from people that are interested in that. And so definitely feel free to reach out through any of those sources if you want to learn more.
That's fantastic. What I want to add is this: yes, we do have a career page. We do our best to put up those ads. We will soon, hopefully we'll have a little bit more time, but we will soon start advertising those more on Twitter.
Oh,
make it more accessible for people. But if you ever have a job description that you want us to put on the career page, happy to do it. Tweet it, tag us, and we'll be happy to retweet the job descriptions as well. And one last thing I wanted to mention, and I think I forgot, but anyway, I invite you also. I know you're giving a live talk at the Dr. GPCR Summit, so I invite you to talk about your lab and as well as the positions that you have at the summit. We expect to have a lot of trainees and a lot of people come and join us for your talk.
Oh, that would be wonderful. Thank you very much.
Of course. So thank you so much, Ben, for your time. I loved learning more about Smoothened, and the cholesterol part blows my mind. And I think even the PKA binding, to inhibit PKA, just mind-blowing discoveries.
Yeah, definitely. And now you can see what I meant when I said that I've learned so much biology from Smoothened. It's really changed the way that I think about GPCRs and all these basic concepts that I thought I understood before. So I'm really glad that you enjoyed it too.
I did. I learned a lot. Thank you so much, Ben, for your time.
Thanks a lot.
Thanks. Bye-bye.
Bye.
I need a favor. Please share the Dr. GPCR University program with your friends and colleagues, especially if they live and work in a developing country. Please subscribe to our YouTube channel. Many of you come back to watch our videos, but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, Attila, Ines, Montserrat, Ivana, Cam, John, Yatsu, Andreina, and Balint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. And don't forget to subscribe to our monthly newsletter on our website to get all the news about all the work that we've been doing and we are doing at Dr. GPCR directly delivered into your inbox. Another great way to support us is to share your favorite Dr. GPCR programs with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com or hello@drgpcr.org. As I mentioned, both emails will be active for a foreseeable future to make sure that we don't miss your email. Until next time, stay safe.